So as Chris mentioned, I'm going to kind of give you an introduction and go into a little bit more detail about some of the genomic applications that we've developed at Labcyte using the Echo Liquid Handler. We're going to be using both the 555 and the 525 for some of these applications. Chris highlighted a few of those and the differences. I'll touch on them as we go through. So for the content of this, I'm going to start off where Chris left off. And this wasn't planned, but it's just how it worked out, and I think it'll work out pretty well. And next, I want to take a little bit of time to talk about our application roadmap. So I want to take the time to really kind of describe the approach that we're taking to demonstrate with context and continuity how you can use Labcyte products in your research. And I think this is important because the roadmap really provides our applications with kind of context. It's purposeful, it's meaningful in terms of what we can deliver and translate that into your research. Because as an application person, I'm really not just interested in transferring or moving liquid. I really want to know what the final solution is. Speaking more specifically about applications, I'm going to take some time to detail a few in the genomic space. I'll touch on what else we can do only briefly. And in those genomic applications I'll touch on, as Chris mentioned, it's going to be synthetic biology. We're going to touch on genotyping, as well as an NGS library prep. And then I'll finish with a kind of highlight feature. Chris already brought him up again, about Christopher Winneberger at FIM, and to demonstrate how he's using the Echo towards personalized medicine. And we can open up for discussion. So as Chris left off, we're looking at miniaturization in genomics. And you wonder why I'd start with a conclusion or a summary slide. And I really don't look at it this way. I mean, honestly, if we look at the trend coming through, it's really a common thread for all our applications. We want to show the miniaturization. We want to show value. And what we've done here is show, in this column here, and I won't go through too many of the details because it's been gone through, are some of the applications that we put in the table, including PCR, NGS library prep, Sanger sequencing, MDA setup, and some of the synthetic biology processes. And what we're looking at is basically the cost per reaction off the shelf. When we reduce the volume, we get a new price, and you end up seeing a cost savings. This makes perfect sense. You know, if you're reducing the volume, you're going to use less reagent. It's going to translate into cost effectiveness, and you're going to save some money. What this normally translates to also is higher throughput in terms of the amount of samples you can prepare, run, test. So when you combine a lower cost and higher throughput, really that translates into higher productivity. So some of the other key benefits beyond the increased productivity are you're using fewer tips. I mean, this is pretty obvious. We're a non-contact liquid transfer unit. We generate less waste with reduced volumes, and we're also reducing the cross-contamination, which is very important in the genomic space. So eliminating carryovers is another key feature, primarily in the drug discovery, but also in genomics. And also we're increasing throughput, as I mentioned. The other benefit, particularly in the library preps, is an increase in your ability to pool and increase multiplex reactions. One thing I'm not going to go into in detail for all of these includes all the areas that are impacted through this miniaturization with genomic applications. And that includes synthetic biology, cancer research, which I'll primarily focus on with personalized medicine, but also drug discovery and academic research, diagnostics even. So as I mentioned, the applications roadmap really should provide purpose. And in our case, rather than sort of a roadmap, it's more of a pathway. And in this case, what we're doing is modeling after the PI3K-AKT mTOR pathway. So I don't know if you're too familiar with it, but essentially what it is you've got extracellular growth hormones. They're going to bind to transmembrane receptor tyrosine kinases. What this is going to do is incorporate PI3K up to the membrane, activate the PI3K. You're going to have it'll catalyze a reaction of phosphorylating PIP2 to PIP3. PIP3 is going to translocate to the membrane, bringing AKT, and that allows PDK to phosphorylate this. The second phosphorylation occurs with mTOR complex 2, and that frees up AKT to dissociate from the membrane, and it triggers proteins and cascades, which are going to include growth, translation, cell survival, and proliferation. So obviously, a signal transduction pathway that's activating cell survival, proliferation, and growth is a targeted pathway that's very relevant to cancer biology. And so this provides a nice tool for us, a scaffold, if you will, to develop applications which can speak to the cancer biologist. The other nice feature about PI3K is it's not just stuck in this one pathway. It's involved in many other fundamental biological processes. So within the RTK superfamily, you also have insulin receptors as well as cytokine receptors. So this is also going to affect motility, survival, metabolism, and intracellular trafficking. The other thing is if we looked at P10 gamma isoform, that's going to complex with the heterotrimeric G proteins associated with GQ alpha and be activated by G protein coupled receptors. That's going to open up a whole calcium mobilization pathway, cellular communication, et cetera. One other nice feature is if we looked into B cell receptor activation, the P10 delta isoform is very active in immune response. So we're able to then develop applications that are going to speak to the immunologists, potential cancer therapy. One in particular could be rheumatoid arthritis. So this pathway is going to provide a scaffold that's going to help us speak to many, many different people. So what makes it so great for cancer biology, beyond simply the pathways that I described, is this pathway is very well studied. There are many, many mutations that have been determined. There's P10 mutations. The IMPP4B is dephosphorylates PIP2 to PIP1. That's another target of interest. PI3K, AKT have mutations. It's very well studied. So what it does is it provides me the tools to provide proof of concept studies for you to relate to in your research. It's also well characterized in terms of the number of compounds that have been developed to treat this pathway. The interesting thing about this pathway is it's brutal. It's not something that stops after one mutation in cancer. There's redundancy. There's excess mutations that pile onto each other, and that leads to combination therapies and ways to interrogate the system, not just with a single inhibitor, but also the combination approach. We're not going to have time today, but this gets translated to different areas in terms of applications that we can provide. Most namely compound transfer. I mean, the Echo is a gold standard in terms of compound transfers. This could be for assay-ready plates, could be for combination screening. It can also be for the ability to direct dose into cells and cellular-based assays. The biochemical assays that we have include binding activation of the PI3K enzyme itself with an FP assay, cell-based assays, proliferation, phospho-AKT. We can even go down into PK-ADME as well as crystallography setup. So it's a versatile scaffold for us to use. Now, to bring it back to genomics, what can this provide us? So there's a very high incidence of P10 and PIK3CA mutations in human cancer. In particular, breast cancer for the PIK3CA, it's 29% and 14% for P10 mutations, and breast cancer at 27% for the PIK3CA. So these are activating mutations. And what they're going to do is provide us tools that we can use to interrogate the system using the Labcyte Echo. In particular, I wanted to focus on a couple of tools that are going to come up throughout the talk. And one of them is the MCF7 cell line. It came up earlier about how cell lines are tough to immortalize post human extraction, and how you can keep them going for a long time. This is one thing that makes this line so relevant, so well studied, is that it's one of the few breast cancer cell lines that has an ER receptor in it. So it's very valuable. It was isolated in 1971. Since then, 25,000 publications have used this cell line. So again, it's very relevant. It's something that a lot of people are interested in. And bit of trivia: the MCF7 is named after the Michigan Cancer Foundation, and it's the 7th try in order to do this. You won't hear the person's name. She was a nun in Michigan, but this line is very important for the study of cancer biology for breast cancer. The benefits to this really is going to be the ER receptor. It's HER2 positive, as well as an activating mutation at the E54K. So that's an activating mutation I'll touch on. This is another example of how the cancer just piles on. There's multiple mutation sites, and it allows for multiple combination therapies. This PC3 cell line is a prostate cancer cell line. And what this cell line represents, it's a little bit more complicated in the prostate cancer, and it reflects a little bit in terms of what we were shown earlier in terms of a progressive state of cancer. If you were to look at the LNCAP cells, they're going to be androgen sensitive. They have the P10 mutation. They're a little bit easier to treat. You remove androgen. Next, you can go to DU145, and then you lose the androgen sensitivity, but it's not tumorigenic. Then you move to the downstream in the cancer development, and you're looking at a PC3 model. Here, it's very tumorigenic, it's androgen insensitive, and it's also P10 null as well as the tumor suppressor P53 null. So we're trying to find tools that are going to be very useful to study and be able to relate back to ultimately what your research goals are. Now, if we look more closely at the PIK3CA gene, because we are talking about the genomics here, we can break it down into a few different domains. It's got the P85 binding domain, a RAS binding domain, C2 domain, the helical domain, where it's going to be folding over itself onto P85, as well as the kinase domain. What we have plotted here are the amino acids and the exons, and here are the 3 points where most of the mutations occur. So in this particular gene, 80% of the mutations occur within these 3 mutations. 2 occur in the helical domain, 1 in the activating in the kinase domain. What this translates to is under normal circumstances, you're going to have a ligand binding to the RTK, and that's going to cause activation. When you have one of these mutations, essentially you have no ligand binding. Rather than being in the inactive state, where it's got a conformational change with the P85, it's opened up, and then it's available to dephosphorylate or phosphorylate PIP2 to PIP3. So this is the mechanism of action by which it's able to create a signal transduction pathway in the absence of stimulation. Now, for the P10, it's got a few more different domains. It's got the PDB domain here. It's got the phosphatase domain. C2 domain is much larger, and a bunch of kind of ubiquitination trafficking domains at the tail. There are several mutations that can occur, and there's a couple of ubiquitization sites. Now, we're not going to necessarily focus on too many of those details for the talk, but just know this is another target that we're able to interrogate. What's happening here in terms of its role is P10 is essentially a negative regulator of PI3K. It transfers back or dephosphorylates PIP3 to PIP2, so it's no longer active to turn AKT on. In this case, you're seeing how one mechanism is ubiquitinated. It's set up for degradation. It can be translocated to the nucleus, or it can just be unstable. In most cases for P10, there's a large mutation. There's 1 to 3 exons that are available, and then it's a large deletion. For LNCAP, the P10 null activation is really just a frame shift. So each of these pieces of information, it's important to know what the basic biology is to then determine the treatment. So the first application I want to step into really is simply qPCR. If it was just qPCR, it's kind of not all that much fun, but this is the standard workflow that we're looking at. You can use a standard liquid handler pipette to do your purification of DNA. You're going to load your samples into a 384 polypro plate. You've got your primer probes in another one, and you can have the master mix in a reservoir. Using the 525, you can mix and match and add all these reagents in a reduced volume, run your PCR, and have your analysis. The benefits were shown on the first slide as well as Chris's last slide. What we tried to do is really kind of push the limits a little bit and make it more applicable in terms of what you might be looking at. So in this particular application note, we're looking at miniaturized PIK3CA mutational analysis. And as you can see, it hits all the common threads you're going to have. You've got the reduced sample input requirement. We've gone from 10 nanogram input down to half a nanogram. You've got assay volume is reduced. This is going from 30 microliters to 3 microliters for the PCR reaction. And quality is not compromised, and I'll show some data for that. So the first step really is to show how you can reduce the sample input in the miniaturized reaction. So in this case, we're taking the control DNA. Using the Echo, we're going to transfer 10 down to half a nanogram per reaction well, and we're going to backfill with water. This is up to 900 nanoliters. At that point, we're going to add 600 nanoliters of our primer probe. In this particular case, it's E54K, E54Q in this case, and then top it off with a microliter and a half of master mix. We're going to cycle it. And basically what we're coming out of, as we're going down through the dilutions, we're plotting the Vic and we're plotting the FAM. And when we're plotting these CP values, we're seeing very good linear relation for both cases at 0.99 in each readout. The other thing is the slope is negative 3.74, which is very good, showing that linearity of the dilution. So from this data, we're able to show we've got a reduced sample input, which is important for all your precious samples out there and patient samples that you might be testing, but also that the Echo is very accurate and precise in its ability to dispense these not only the reagents, but your sample itself. Now, the next step is really a question about the sensitivity and the quality generated by the Echo. So in this case, we wanted to check the limited detection at reduced sample input in the miniaturized reaction. So again, this is going to be a 3 microliter reaction, and what we're going to be doing is looking for the detection of a particular mutation. We looked at 5 different mutations. In this case, I'm just showing the E54K. It's the activated mutation in the MCF7 line. And what I did was essentially combine different combinations of mutated DNA as well as wild-type DNA to get different percentages that are available to this. Testing the 3 different inputs, we run our CP values and measure VIC and FAM. So as a reminder, VIC is going to be essentially the DNA present. FAM is going to be the detection for the mutant. And we've got the criteria here for what's going to be a positive signal. At 10 nanograms per microliter, we're below even the 25 in some cases. So effectively, at the reduced volume, we're overloading the system. We can't use that much DNA, which is a good thing. You want to use less if that's possible. In the graph over on the right, we're plotting the reaction curve at 1.25% mutant amplification curves. And in this case, we've got the 3 nanograms with an average CP of 26 and the average CP for FAM at 37. So this is going to be a positive call using 3 nanograms per microliter, or 3 nanograms per reaction in the reduced 3 microliter volume. This would be equivalent to the kit recommendations. So again, we're showing that we're not reducing any of the quality data that you can generate from reducing the input amount or the reaction volume. Now, it's finding good with control DNA. The next step really was to show, does this really work in real life? So taking the MCF7 cell lines, we went and prepared the DNA. In this case, we're using the BioLine Isolate 2 genomic DNA prep. We're taking this prep from the MCF7 cell line, and we're going to do a cross-test against all the different available mutants in the kit and look to see for positive calls. So again, we used a 10 nanogram total input and 3 nanogram total input. And we looked for positive responses across the different specific primers, which would detect mutations. So in all cases, they were not called. There's no mutation present in the MCF7 DNA, and this is as expected. We know this is a control line for E54K mutations. And we can see here on the left side, both at 10 and 3 nanograms total reaction, that we are getting positive calls, robust positive calls in the reduced sample. So this is very good for us. We're getting correct calls at the reduced reaction volume and reduced sample input. The next is you also don't want false positives. So same system was set up and run. We're looking at the PC3 cell line. So this is known to have the wild type PI3K in there, and we're not getting any calls as positive for any of the mutations. So not only are we getting good calls for our positive control, we're also not getting false positives from negative control. Next step is looking at synthetic biology. So in sort of a traditional workflow, you're going to have your oligos on 384 well plate. And what you can do here is actually get these pre-plated from Twist or IDT to make it a little bit more convenient for you in our Echo source well plates. You can run a PCA reaction or MDA, whatever you need to do to amplify the DNA. You can also swap that out for using the Gibson Golden Gate. There's a variety of ways to amplify that DNA and stitch it together. You run the fusion and QC offline to test and go into your cloning system. This can be bacteria or into mammalian cells. Offline, you're going to be pretty much running your transformation of bacteria, growth, isolation, et cetera, and then you can come back and confirm what you've built in your synthetic biology platform, looking at an NGS library prep. So I want to highlight a little bit about one of the reactions of the 3 that we have developed protocols for, and this is going to be the Gibson one. It's probably the more popular for piecing together in gene assembly. And what you've got essentially is your vector, and you're going to put in your different insert DNA. These are the ones you want to piece together and stitch to make a longer fragment. You're going to require a certain amount of overlap in case of these inserts, so your design's important. In a single tube, you're going to add your pieces of DNA, your plasmid. You've got your 5' exonuclease, and then you've got your polymerase and ligase. So what's going to happen in that well is you're going to chew up the 5' end. You're going to have overlaps. Those overlaps are going to anneal, fill in the nicks, ligate it, and you now have an extended piece of DNA. Now, in the case of real-life application, one thing that's important is, as I said, multiple mutations can occur within the same system, at least in breast cancer. So if we wanted to really interrogate this system further, we're going to be looking at different systems that have both the E542K as well as H1047. This is a really fast way to piece together this piece of DNA and that piece of DNA, mix and match your mutations, and then you can develop protein. You can test a biochemical assay for inhibition. You can even transform and create new cell lines. So utilizing the Gibson assembly, it gives you a lot of versatility. And thinking about the AML study where they had the massive amounts of mutations, this might be one route where you can sort of pick and choose which are the most important mutations to look at. Of course, it depends on if it's a particular gene of interest or multiple mutations with a gene, but it's sort of a fast way to help prioritize potential work. What I've done is taken the cutout or the tables from a paper that Paulina Kanagowski wrote in Patrick Kai's lab in the University of Edinburgh. And this is in the JALA article here, where it's smart DNA fabrication using sound waves. So what they did here was run the Gibson assembly, and what they want to do is compare the manual reaction by doing a stepwise reduction in the total volume of the echo. So as you can see here are the mixture of reagents that they're adding, and in a total reaction volume of 20 microliters, they're able to reduce the total reaction volume to 50 nanoliters. What they've done is plot their results here. I believe this is the cost on this axis. Over here is the efficiency, and they're plotting it relative to the total reaction volume, and the manual here is control. So their takeaway really was they got correct assembly. It was observed as low as 250 nanoliters, and that was a 20-fold reduction in cost to them, and that there was no background as 100% correct assembly. Now, I won't go into even more depth of this, but this paper is a very good resource if you're looking into these types of reactions. What they also did was profile the Golden Gate assembly reaction in a similar fashion and had similar results. So it's, again, a reference for you to look at that chemistry if that's your preferred chemistry. They also provide a good reference for their miniaturization of the PCR reaction. Now, in terms of synthetic biology using the liquid handler and the applications that we've developed, what I did was essentially use the Echo Liquid Handler and the gateway gene assembly. Golden Gate was done, Gibson was done, used gateway in this particular case, and did that to generate and transfect a P10 GFP construct, and I put that into the P10 null PC3 line. This was very straightforward, and it was a bit of a shortcut method. It's not going to be your traditional route, but it certainly can be if you're looking for sort of phenotypic and quick answers. So in this particular case, I just took 6 microliters of the entry plasmid. This entry plasmid I created with my P10 gene. I put it in with a 3 to 1 ratio to the destination plasmid, and that destination plasmid had a GFP as well as a selectable marker on it, and used the clonase 2 reagent to do the swap out. The reaction was run in a 96-well plate. To that plate, I added the transfection reagent, assuming the reaction went to completion was high enough efficiency, and then transfected that onto cells. One point to note, if you're doing many transfections, you know that transfection is not exactly an easy, straightforward method to do. You got to worry about DNA concentration, your amount of lipid amounts that you're going to do, sensitivity to the cells. So what I've done in the slide, and you're welcome to these, are a couple of references that highlight the ability of the Echo in order to run those matrices for you to determine the optimal transfection combination or the optimal conditions to get a transfection efficiency within your particular cell line. So once I had the phenotype, that was a GFP expressing line. I knew in cells I had a GFP expression. One question I had really was, well, I know a construct's in there. There's a small chance that the destination vector didn't open, and all I'm doing is selecting for GFP in there. So I wanted to make sure that the gene of interest was present. I'll go into this next, but looking at NGS as a QC, we did the library prep from this particular cell line and ran a real low coverage in order to determine if that open reading frame was present in the DNA. So here's the IGV plot, and I've overlaid the previous graph that was shown earlier for P10. We have the 9 exons here. And in general, we do get coverage. 95% of it is covered at 5%. And overall coverage was 10%, I believe, and a portion of it was not well covered. So my next step here is going to be take this cell line, put it in some of our functional assays to make sure we actually have functional protein. So we've gone from essentially the synthetic biology of creating a construct of a gene system, did a transfection, created a recombinant line, QC'd it with the NGS library prep, and we're going to put it into functional assays for cellular pharmacology and make sure that the cell in the construct is functional. All of this was done with the Echo. I mean, not entirely, but the Echo enabled each of the steps. So talking about the library prep, what I'll do is focus on the Nextera kit in this particular example. The XT is very similar to this. In general, the Echo prefers the fragmenting enzyme preps. This could be the Kappa, the NEBs, Nextera, et cetera. But it doesn't, you can also be using your Kavaris-generated fragments if you'd like. That would be done on the offline liquid handler. You put it into the PCR plate. With the Echo, you're going to add your TD buffer, the ATM buffer, run the quick PCR, add the NT or SDS. I'll focus on that in a second in terms of the ability to develop the protocol for that. And then you add your master mix and index. So you're going to create your entire tagmentation reaction with the Echo. That's going to go off to amplified DNA, and then you're going to do the cleanup offline with the beads typically. When you do the elution off the beads, you can take that back into a source well plate and do the quantitation and normalization with the Echo at that point. So this will be an applet that's coming out shortly, and it's going to be a method to streamline miniaturized Nextera library preparation using the Illumina next-gen sequencing. And again, we're using this recombinant line that I generated. We miniaturized the reaction for the tagmentation at least 20-fold without sacrificing the data quality. We also reduced the PCR reaction, so this was down to 5.5 microliters from whatever you might be running. And we increased the throughput really because we switched it from a 96-well process to a 384-well process. So the higher density is going to enable a little bit higher throughput, fast processing of your samples. All in all, this leads to a reduced cost in your reagents. So the assay design and setup really focused on 4 different libraries that were used. So within the Nextera kit, there is a Zymo cleanup step, and the Zymo cleanup step is a spin column. So it's not very automatable. What you want to do is try and eliminate that step so then you can run it a little bit more streamlined. The Zymo step is really meant to strip the DNA of the fragmentase or the tagmentase or deactivate it. There was a paper in 2015, Pacelli, I think, and they detail the method to use SDS as an equivalent reagent to handle that tagmentase on the DNA. So the 2 reactions that focused on looking at included the Zymo cleanup were run at 25 microliter total volume and also 25 nanograms DNA, which was half the manual or the protocol that's described. The Zymo step was also then reduced to 5 microliter total tagmentation, but in this particular case, the PCR reaction was kept the same. The next 2 reactions to compare were using an elimination of the Zymo process for the streamlined process and transferring 0.1% SDS. In this particular case, we went straight to a 5 microliter tagmentation reaction, scaled appropriately, and had a 10 microliter PCR reaction. And then we further reduced with the SDS treatment to 2.5 microliters tagmentation reaction and further reduced the PCR reaction to 5.5. As an example, this is the library that was generated from the SDS 2.5 day library. This was taking 1 microliter out of the final library, run on a tape station, and the electropherogram is plotted here. Now, one point that was brought up in the general workflow was the importance of DNA quantitation. And it's very important before it goes on the sequencing, but it's also important to have the amount of DNA known before you go into the reaction. So being able to quantitate it accurately is very important. So looking at the input genomic DNA, I'm going to show the example of using the Promega QuantiFluor 1 DS system. It's a fluorescent readout. It's similar to picogreen, and how well the Echo was able to handle this. So on the plot here, I'm looking at the fluorescence versus the nanogram per microliter on a log scale of the control DNA, K562. And we're seeing very good linearity at 0.99. When I wanted to take my sample that was prepped, in this particular case, the prep came from a pure gene kit. I wanted to find out what the concentration was and how well it transferred. So in this particular case, I'm looking at the calculated nanograms per microliter, which is taken from these fluorescent relative to the control versus the expected, and this is coming from the vial of the tube. So that also worked out pretty well in the 0.99. There's a slight deviation off center, which might just be the initial quantitation from the vendor. It may be in my hands. But the linearity is what's important in terms of showing the accuracy of the transfer. And there are error bars on there, so the DNA transfer from the echo is quite precise. Now the person left who asked earlier about fragment or potential damage to the DNA, and here the electrophotogram is showing very clean and a normal distribution around 13 kb size DNA. So we're not seeing the fragmentation that the question was brought up. It's a common question, and oftentimes people do assume it's sonication versus a low frequency acoustic transfer. So that's what this was presented for. Now, when we look at the sequencing results, we are seeing comparable results. Now, the samples were pooled. There's 4 put together. We're run on NextSeq for the 150 base pair 300 cycle kit, and we're looking at some of the metrics that come out of it. So in terms of the percent aligned, the quality of the DNA, or the quality of the reads for the percent Q3 in the mid-90s, good percent alignment at 92%. Remember, this is whole genome sequencing, so there's quite a bit of extra DNA. Oftentimes you might see in plasmid sequencing that value at 100%. That's realistic. In whole genome sequencing, we're a little bit lower. Other metrics are in line with what we expect. The GC content, the literature value is 44%, and there's a slight discrepancy in the coverage, which we can look into in more detail when we compare the Zymo versus SDS. So I don't have a good explanation for that, but it's something that we can look into more. Down here are some of the plots that are generated. In the bottom left here, we've got the Q30 score for the 2.5, the SDS 2.5 reaction. We have very good reads here. Here's the duplication plot and the GC content. So we're getting good distribution, not a huge hint of PCR bias and duplication from that route. So we're pretty happy with having developed a next-gen system library prep where we've reduced the volume and automated or streamlined the automation system. Sorry, Randy wanted me to go slower. So in terms of personalized medicine, I wanted to highlight some work done here at the Finnish Institute of Molecular Medicine, and this was Christer Wennerberg's work that Chris highlighted earlier. And their approach is to use a database research and identify potential drugs and resistance profiles. So they're taking a bit of informatics approach, a little clinical history approach, and then screen the repurposed drugs and novel drugs combinations and characterize the resistance. Now, the drug sensitivity and resistance testing was enabled by the liquid handlers. Its ability to do the combination screens at low volumes made all of this work possible. So by identifying the patients without resistance, they're able to treat them with the correct drug combinations. So a lot of the information that goes into this is in terms of the genome, epigenome, as well as transcriptome of the patient. A lot of data is being generated around them, both pre-dosing, and they're also following up with the patients afterwards to sort of track progress post-treatment. And the results, they've gotten complete remission in morphologic leukemia-free states in more than 40% of the cases that they've tested. Several approved drugs can be repurposed for certain AML patients. So they've had some great success using the Echo to sort of enable their efforts toward personalized medicine. Over on the right here is essentially the cycle that they're going through in terms of their individualized system medicine program. And as you can see, the design treatment is something that they're looking at from an informatics perspective. They're taking all the data possible from the clinicians, from their personal profiles, et cetera, and they're designing the DSRT program for them. Treat the patients, they take the genome transcriptome, and compare the results. As you go through this cycle, you can see here that the Echo really does play a role in a lot of these different steps where the testing occurs. So you've got the study of the biology of the disease. This is what we're trying to accomplish by having the roadmap and show where we can apply the applications. Here you've got the mechanism of action. It's another thing the Echo is able to help you with. The patient specificity with the DSRT and the treatment, he's highlighting very well how he's using the Echo to enable this. And now with the NGS library prep and soon RNA-seq work, the genome transcriptome profiling will also be enabled by the Echo in his lab. His general approach is to plate the compounds with the Echo up front, have these assay-ready plates, again, in the combination studies. He's using cherry-picked various combinations, and he's also doing antibodies and siRNA. Adding cells similar with the multi-drop, they incubate overnight, and they're looking at essentially a similar cytotoxicity assay, looking for sensitivity to the combinations of the various cell patient samples tested, and that goes through to the analysis. And I'll end with the slide that you've already seen, which is what Christopher is saying about us. So essentially he's saying that the Echo is very useful in his studies and is appreciative of its use.